right. Well, I'm going to start the webinar now. My name is Jenny Kimball. I'm the managing editor for the Journal of Molecular and Cellular Cardiology. Thank you, everyone, for joining us this morning or this afternoon, wherever you may be. This is our next webinar in our Editor's Choice series, and we are pleased to have Mike Shattuck join us as the moderator for this session. So I am going to pass it over to Mike now, who will introduce our speakers for the day.
Thanks very much, Jenny, and welcome everybody to this webinar. It's a great pleasure for me to host this. I haven't seen Mark for a while, and I know that he's always got some really stimulating and interesting stuff that's bubbling along. This particular paper was published earlier this year. Just by way of introduction, I should say that I've personally been interested in excitation contraction coupling and calcium regulation now since I went to University College in about 1980 as a visitor to try and work out how on earth to measure calcium in a cardiac cell. And I met up with some of Mark's old compatriots there, David Allen and David Eisner and people like that, who showed me how to go about working with cardiac muscle. And as a young, fresh out of university graduate, I hadn't a clue what was going on. And I've watched with massive interest as Mark has pretty much defined many aspects of excitation contraction coupling in terms of understanding ryanodine receptor function, calcium release channels, how they come together to produce the calcium transient, the spatial geometry of the problem, the mathematics of the problem, and indeed developing techniques which just are mind-bogglingly clever to try and understand how EC coupling works. And so it's a real pleasure for me, rather towards the end of my career rather than the beginning of my career, to actually hear what the state of the art is. So Mark, thank you very much both for your years of illuminating me in the dark arts of EC coupling, but also for being here today with Cherrie. And I know you're going to tell us something very interesting about your paper published earlier this year. So Mark, if I could hand over to you, if you could share the screen. We will have a question and answer session at the end, but if anybody wants to post comments or questions in the chat, we will deal with those at the end as well. So Mark, over to you and Cherrie.
Great, thanks. Can you see my screen?
I can.
Okay, great. Well, thank you very much for that generous introduction, Mike. And as I haven't seen you either for quite a while, although I know you've been busy beavering away over the years as well. And I'd just like to acknowledge Cherrie Kong here beside me, who did most of the work on this paper, which you've alluded to at JMCC. So the starting point is the title of the paper. We were interested in calcium spark latency and trying to understand what controls the latency in the calcium release event in cardiac ventricular myocytes. Now, by way of introduction, I'm going to give you a little bit of the background of the thinking that led up to this paper, and then Cherrie is going to take over and give you some of the fine details and some of the conclusions from the paper, and then we'll maybe wrap up the end and have some questions and answers. So we've known for a very long time that excitation contraction coupling occurs at the junctional SR, the JSR, those are the terminal cisterns of the sarcoplasmic reticulum, where they make close apposition to the T-tubules, which carry excitation into the working ventricular myocyte. And if there aren't T-tubules, then there are similar junctions on the surface membrane of the cell. And it's at this junction that the key machinery is placed. And these release units, calcium release units or couplons, were described by Clara Branzini Armstrong quite a long time ago. And this is from a review of hers. And we can see in this high-power electron micrograph various structures. There is the sarcoplasmic reticulum, the major calcium store inside the cell that is triggered to release calcium. It is triggered by a calcium current, which develops in the surface membrane of the cell. It was first described by Bieler and Reuter some 50 years ago now. And we now know that that calcium influx takes place from this space via L-type calcium channels. And that calcium influx enters the dyadic space, the space between the T-tubule and the SR, where it activates calcium-sensitive channels called ryanodine receptors, which are embedded in the sarcoplasmic reticulum membrane and are the calcium release source of the sarcoplasmic reticulum. So our view of EC coupling is summarized in this cartoon here. You can see on the left side T-tubules with their dihydropyridine receptors, which are L-type calcium channels. And they allow calcium to come into the dyad space. And in that space are also these green ryanodine receptors that pick up that calcium and release even more calcium. And in that space, the calcium levels are very different from the bulk calcium because it's restricted. And there's all sorts of things like electrodiffusion taking place, which we sort of know a little bit about. Of course, the SR, as a store, has to take the calcium up that it released and for a steady state. And that's done, of course, by calcium pumps. But we're not going to be talking at all about calcium pumps. We're just going to be focusing on the release event triggered by dihydropyridine receptors, trying to understand what controls the latency and the voltage dependence of this process. So we know that the ryanodine receptor and the DHPR are the major players in this machine that releases calcium. But we know much less about their exact localizations. And some of the best work in attempting to localize these proteins has been done by Ed Moore and his group. And here you can see a high resolution image of some immunolabeling of dihydropyridine receptors in green and ryanodine receptors in red. And where they co-localize, you see white. So you can see that a lot of the junctions have whiteness in them. So we know there's co-localization, but it's not at all clear what the stoichiometries of these proteins might be, for example, or exactly how they're organized from such images. And although we had tried to quantify the number of ryanodine receptors quite a long time ago on the basis of the size of these blobs of ryanodine receptor labeling, it's something that's actually extremely difficult. I mean, better methods have come along, which I'll talk about in just a moment. If you try to improve the resolution by using electron microscopy, then in principle, you might be able to see where these proteins are. And on the left, you can see here's some images of freeze fracture by Clara Franzini Armstrong in her review in 1999. And she ascribed these particles in the membrane seen here to be dihydropyridine receptors. Although, of course, we can't be sure that they're dihydropyridine receptors. It's a freeze fracture with some lumps in it. But from those images, she suggested that the dihydropyridine receptors were loosely placed over the surface of the underlying junctional SR membrane, where the ryanodine receptors in the form of tetrads might be packed into an ordered array. So although in skeletal muscle, we seem to have a very regular organization of these particles in the membrane, in cardiac muscle, that regular organization was lost, and it's not really clear how they are located with regard to attempting to localize the DHPRs with immunogold, here a paper by Gathical, illustrates the big problem with trying to identify individual proteins in the EM. If they're not big enough to see by yourself, immunogold is extremely difficult. And you've got a very large particle being tethered by an antibody onto the presumed locations of dihydropyridine receptors. And you can see the labeling density is very low. So does this mean that there's only one or just a couple of dihydropyridine receptors per junction? Or is this picture where there's a lot more? Is this more correct? So we're not really very sure what's going on here in terms of stoichiometries and exact localizations of DHPRs. Using biochemistry, we can look at the total concentration of receptors. And here we can compare ryanodine receptor binding and PN200 binding, PN200 being a dihydropyridine. And we can see that it seems to be variable amongst different species, varying from about 4 to about 10 to 1, depending what species you look at, apparently. And here we'll just pick rat because it's our favorite animal to work on. And here we see an overall binding ratio of 7 to 1. So 7 times as many RYRs as DHPRs. But of course, this again might not be the whole story because we don't know exactly where those DHPRs are. We don't even know where the RYRs are. And to underscore that point, of course, we know that not all RYRs are in those junctional terminals in the dyad. There's, for example, corbular SR, where the RYRs are not linked to the T-tubal membrane. So this sets some bounds on total protein, but may not tell us anymore about detailed stoichiometries and localizations. So from functional studies, some statements have been made about how things might be organized. And we suggested a very long time ago, me and my collaborators, John Leder and Luis Santana and others, that a single L-type calcium channel could activate a calcium spark. So that's a lower limit. I suppose at one level you might say it's intuitively obvious that a calcium channel could, but the evidence was actually quite strong at the time that at least at negative potentials, the coupling seemed quite tight between the L-type calcium channel and sparks. However, at more positive membrane potentials, where the calcium current is smaller, it was suggested that you needed a large number of well-synchronized L-type calcium channel openings to get an efficient EC coupling process. That was suggested by Altamirano and Burrs in 2007. And Peace Chang attempted to measure, with his group, attempted to measure and link the L-type calcium channel directly to the activation of spark with a loose patch technique. And they modified L-type calcium channel gating with FPL 61476. And with that, they get very long L-type calcium channel openings. And you can see here an L-type opening very strongly correlated with the nearby calcium spark. And from these data, they suggested that an L-type calcium channel might trigger about 4 to 6 ryanodine receptors to generate a calcium spark. But of course, here we're dealing with modified calcium channels whose influx is going to be perhaps much larger than normal because the gating has been modified. Chang and Leder in their review from published data suggested that only about 1 out of 60 L-type calcium channels could activate a calcium spark in that diet. In other words, a very low coupling efficiency between the L-type calcium channel and the RYR activation, and that's at 0 millivolts. And a similar conclusion was reached by Polakova, suggesting that a high number of DHPR openings is necessary to trigger the release unit. So there's some quite strong statements obtained by extrapolating data from very negative potentials or from modified calcium channels. But it seemed to us that this is a little bit of a problem because if the opening probability of the DHPR is quite low, as it appears to be, then very large numbers of DHPR openings would be required to activate the calcium spark. And that would imply a very long latency compared to the close time of the L-type calcium channel. So that could be a problem because we already knew from our very earliest work using high-speed calcium recordings that EC coupling took place inside a few milliseconds. So how do you get something happening in a few milliseconds if you require hundreds of openings or lots and lots of L-type calcium channels? Quite a long time ago, we considered what might happen in the diode when an L-type calcium channel opened, and that's shown here on the left, some calculations incorporating electrodiffusion. And the point I would make here is that when an L-type calcium channel opens, very high calcium levels are generated extremely quickly. So here we see the green color is about 10 micromolar calcium. And those high calciums are required to get relatively calcium insensitive ryanodine receptors to open in a timely way. In the subsequent paper to that, we actually looked at RYR activation, and we didn't know very much about RYR activation at that time. We assumed a diffusion limited on rate, for example. And here you can see that it was clear, because the plume of calcium is quite restricted within the dyad, that the geometry of the ryanodine receptors plays an important role. And of course, at this time, we were led by Clara Franzini Armstrong's ideas about how tetrads might be close packed together. So we looked at various packing organizations. And the interesting thing here is that you can see that the cluster activation is higher for one ryanodine receptor on its own than if you have two ryanodine receptors side by side. And the reason for that is because of the gradients of calcium being transduced by the ryanodine receptor. More recently, very high resolution imaging methods have been developed, such as DStorm. In principle, can see single channels. And here's some work. And you can see here from the high resolution DStorm images, various clusters of ryanodine receptors. And within those clusters, we could put ryanodine receptors in a close packed array and estimate how many ryanodine receptors there might be there. And this is important because it's been suggested the cluster geometry is variable depending on the state of the animal, whether it's disease, beta stimulation, etc. However, clusters within the cluster, ryanodine receptors may not be close packed. And here's some data from ISHR showing some very high resolution tomographic data from EM, showing that ryanodine receptors are not in fact close packed and have variable arrangements in their work, depending on, for example, magnesium levels. A technique that gives even more photons per receptor is DNA paint. And with that, Jai and colleagues have been able to identify the positions of individual ryanodine receptors within the junctional complexes. And here we see a cartoon on the left of different complexes. And on the right, we can see the number of RYRs from the calibrated DNA paint data against the apparent cluster area. Draw your attention here to this line, which I've put on their figure, which shows what the relationship would be if the ryanodine receptors were close packed. So clearly they're not close packed, and the work of Cannel and Sola quite a while ago showed that therefore you've got to be a little bit careful about how you put ryanodine receptors into the cluster in order to be able to interpret their gating in any model or from experimental data. So this led us to some questions to think about that we hopefully have addressed in our paper. So if many L-type calcium channel openings are required to activate a spark, how does that relate to the short latency observed for excitation-contraction coupling of about 2 milliseconds? And if the ryanodine receptors are not close packed, how do they transduce the local calcium channel openings? What's the coupling efficiency? And what is the stoichiometry between active L-type calcium channels and the local group of RYRs opposite that L-type calcium channel? And importantly, what's the fidelity? In other words, what's the likelihood that an L-type calcium channel opening, even though that will be exponentially distributed in duration, what's the likelihood that it would trigger a calcium spark? And finally, how much of the natural variance in calcium spark timing is how much of that variance is related to either the gating delay in the LCC or the gating delay in the ryanodine receptor? So the question is, what might we deduce about excitation-contraction coupling by measuring calcium release latency? And by analogy of the utility of single-channel recordings, here we're doing effectively single L-type or cluster recordings, recording calcium sparks or transients or whatever. So the idea is that we have a latency in all these steps, and that inside the observed latency is the latency of all these individual processes. So to get the firm data that we need to start describing how the individual latencies work together, we need to measure latency in excitation-contraction coupling. And that is the work that's being done by Cherrie, who's with me. And she's going to tell you a lot more about our measurements. So take it over, Cherrie.
Okay. So I guess my voice is okay. So we started off by measuring calcium release latency during an action potential by taking ventricular myocytes from rats, voltage clamping them to an action potential waveform. And that waveform is shown in the inset here. alongside the corresponding nifedipine sensitive current. Now, the same data from that same cell is shown in the main figure at a slower time base, alongside the line scan image of calcium release. And the high spatial temporal resolution of this image allows us to appreciate that calcium release has a variable delay at different Z lines. And we can show this again down here. The black trace shows the spatially averaged time course of calcium release, while the colored traces show the time courses at the indicated locations above. And so the observation to note here is that the time courses are very similar at different locations, with the difference that they start at different times. So while this data suggested that calcium sparks may be activated with variable delays during the action potential, this is quite difficult to measure during the calcium transient because calcium spark probability is so high, and because they overlap. And so to overcome this problem, we applied nifedipine at a high concentration to block most L-type calcium channels and thereby reduce P-spark dramatically. So the result of this maneuver shown here, this is the result of 4 sequential AP stimuli with the upstroke of the time of the upstroke of the AP shown by the red arrows. And these data show that for a given AP, that calcium sparks occur at variable delays, and that sequential stimuli will cause a different pattern of excitation. So now from these data, we can measure the latencies of each calcium spark, and the histogram is shown in red in D. The mean latency for these calcium sparks was around 9.5 milliseconds, which corresponds to a membrane potential of around -15 millivolts during the AP. We can now also compare the situation to the case when nifedipine is absent. So during these calcium transients, we measure the latency to calcium release at the Z lines, and these latencies are plotted in blue. And the average latency in this case was around 5.5 milliseconds, which corresponds to a membrane potential of about 0 millivolts. So we want to know what controls this latency and this distribution, this latency distribution. So to try and figure that out, we measured the voltage dependence of calcium spark activation during a depolarizing step protocol. So in this protocol, as shown in the schematics at the top, we simply depolarize the cell to varying step potentials. And these potentials then activate calcium sparks, shown by these images here. And hopefully you can see that the rate of calcium spark activation appears to be voltage dependent, as is the latency, as is their latency. So we can plot the voltage dependence of calcium spark rate in B, and that shows a bell-shaped voltage dependence, as has been shown before. And we can also compare this to the case when nifedipine is absent. So we also now here plot the voltage dependence of the maximum rate of rise during calcium transient, which is a proxy for calcium spark rate. And the similarity in the voltage dependence between these two curves suggests to us that the application of nifedipine, while it blocked calcium channels, did not markedly change their gating kinetics or the behavior of AC coupling. So now we can examine the voltage dependence of the calcium spark latency during DP, and we found that this relationship was U-shaped, with a minimum occurring at around 0 millivolts. And so this U-shape sort of supports our idea of what is happening during this activation protocol. During DP, we are activating L-type calcium channels and ryanodine receptors. So this calcium spark latency should reflect a combination of the two latencies. This is supported by the shape because the rate of L-type calcium channel activation decreases with increasing step potential, while on this side, it possibly reflects the increasing delay to ryanodine receptor activation with increasing potential. And this increases not because of a direct voltage dependence of RYR gating, but that unitary calcium current is smaller at positive potentials, which translates to a smaller or a lower calcium concentration in the diode and a lower rate of activation of RYRs. Another point to note is that the distribution of the calcium spark latencies at any given potential is right skewed, and this is reminiscent of the distribution of early and late calcium channel openings. Now, since this this latency relationship seems to depend on both LCCs and RYRs, we wondered whether we could remove or minimize the impact of L-type calcium channel gating to calcium spark latency. To do this, we used another voltage protocol where what we called the repolarizing step protocol, or RP. And in this protocol, we first step the cell to plus 50 millivolts to activate the L-type calcium channels, but prevent any calcium influx until repolarization to the test pulse. And during this protocol, you can see that again, calcium sparks are activated stochastically, and that spark rate and latency appear to depend on voltage. And when we plot this out, we found that the calcium spark latency during RP increased monotonically with voltage, supporting the idea that we have removed from calcium spark latency the latency to the first opening of the L-type calcium channel. Now, when we're thinking about these different protocols that we've applied to the cell, we thought that during RP, because we removed one source of variance, that is the timing of L-type calcium channel opening, that the variability in calcium spark activation may be reduced. And this is, there is a trend of this, of RP, the variation of calcium spark latency being smaller in RP compared to DP. However, what was surprising was that when we measured the coefficient of variation during AP, it was much, much smaller than in any of the other two protocols. This is surprising because during AP, you still have the stochastic gating of L-types and RYRs, as well as the added variability of the action potential changing over time. So to understand why this is and to answer the questions that Mark proposed earlier, we developed a detailed computer model of HCC. So this model includes various processes, some of which we control. So of course, VM, we control the form of that. And then L-type calcium channel gating, RYR gating, and how calcium develops in the diode was calculated using data from published studies. This model then calculates all how these events happen, and then calculates calcium spark latency. So our model calcium current has a number of features, including voltage, calcium, and time-dependent gating kinetics. And in panel A, you can see that the model can reproduce whole cell calcium transients, calcium currents, and that's shown at various step potentials there, test potentials. It can also reproduce the current voltage relationship as shown in B. It was also able to reproduce the open probability shown in C and the off rate shown D, reported by Joseph et al., who measured these single channel properties and near physiological conditions. But it is important to note that we couldn't find any data at higher potentials. And so we've extrapolated the relationship depending on what other channels have done. To model the RYR, we took data from 2 studies that looked at calcium RYR gating and rat channels. I've plotted there on the top right here their on rates from the 2 studies, and our model uses the formulation that's shown in red, which is between the 2 published studies. Our RYR organization was taken from the EM work of Cherrie et al., and that's shown, the channels are shown here, but we've also overlaid a distance map from the channels to indicate where locations of L-type calcium channels may be. To calculate how calcium develops in the dyad, we set up a model that incorporates electrodiffusion and buffering effects. So we calculated what happens to calcium when it's affected by sarcolemmal and SR membranes, as well as when other charged species apart from calcium are also affected by charge and diffusion effects. So on the left, you can see how calcium in the dyad accumulates from a single L-type calcium channel opening that's open for 1 millisecond. The things to note here are that even though influx of calcium is instantaneous from time 0, calcium still takes sub-milliseconds to develop. And if you look at the different curves which reflect the distance from the channel pore, you can see that the calcium gradient is quite steep and that the calcium concentration drops off quite rapidly. These colors also correspond to the distance map that I showed earlier, and I've placed it above. So this figure shows how the model works. So this is simulating the situation where the cell is being depolarized to 0 millivolts. And the top trace shows the membrane potential, also showing that the model takes into account clamp speed. Then the second trace shows L-type calcium channels gating stochastically, and these then relate to calcium influxes. These calcium influxes then cause an increase in calcium concentration in the dyad, and the colors correspond to the locations indicated by the inset. Things to note here are that the non-linear response of calcium, of the dyad to calcium influx, means that very short calcium openings do not confer a high level of calcium accumulation, while long openings do not increase calcium much further than a similar but shorter opening. This fourth trace shows the ensemble on rate of the ryanodine receptors in this cluster, and this shows that this, along with the bottom trace, shows that even though the RYRs are responding to calcium, because it's stochastic, in this case the CRU has only opened at 15 milliseconds. So in this simulation, the calcium spark latency is 15 milliseconds. So we use this model to now look at the calcium spark latency data that we obtained using the RP protocol. So this graph shows the experimental data on the left at 3 different potentials. And we chose these potentials because they encapsulate the potentials at which most calcium release occurs during the action potential. The lines show the calculated calcium spark latency at the different potentials at increasing RYR to LCC stoichiometry. So by comparing the experimental to observed data, we can see which stoichiometry can best explain the observed data. So for example, at 15 millivolts, a stoichiometry of between 3 and 6 can explain observed latencies, while at more negative potentials, a stoichiometry of between 4 and 8 can explain the stoichiometries. So together, we suggest that at 3 to 8 RYRs per LCC can explain the latencies we've observed here. And we've termed this unit the initiating complex. Using either a stoichiometry of 4 to 1 or 8 to 1 RYRs per LCC, we were able to reproduce the calcium spark latency during RP, as shown in C, as well as the voltage dependence of calcium spark rate during RP. So I didn't show this experimental data before, but just want to point out some features. So during RP, at positive potentials, the calcium spark rate decreases. And again, this supports the idea that at positive potentials, the unitary calcium current is reduced, so it's less able to trigger calcium sparks. What was initially surprising was that calcium spark rate also decreased at negative potentials. And this seems counterintuitive because the unitary calcium current is very large there. However, this is due to the voltage dependence of L-type calcium channel gating. So both PO and open time is decreasing with decreasing membrane potential. So now that we have a good agreement between the experimental data and the computational data, we can examine the computational data more closely to see if we can gain more insight into the details of EC coupling. So if I look at every simulation and look at the calcium channel openings that occur prior to calcium spark activation, I can get an idea of how many channel openings are required. So A shows the fraction of calcium sparks that were activated for N number of calcium channel openings. So for example, the pink shows that the fraction of calcium sparks that required 1 calcium channel opening to be triggered decreased with increasing step potential. Then from these data, we can calculate the coupling fidelity, or P-couple, the likelihood that a calcium channel opening can trigger a calcium spark. And this decreases with increasing membrane potential. Again, supporting the idea of the reduced unitary calcium current being less able to trigger calcium sparks. Now, if we look at P-couple during the voltage range where most calcium sparks occur during the AP, we can estimate that P-couple during an AP is probably around 0.5 or between 0.4 and 0.6. But to get at this from another angle, we input the experimental AP waveform into our model and use this to generate calcium release latencies. We did this for the case of 1 initiating complex per coupon, that's in red, as well as 4 initiating complexes per coupon. And we found that this arrangement could reproduce the experimental data that I showed earlier. I'll try to overlay them now to show their good agreement. And when now I look at these data more closely, I can see that indeed P-couple was around 0.5 during an AP. That is, on average, a calcium spark occurs at the second calcium channel opening. One thing to notice is that the reduction in calcium latency between the calcium sparks and calcium transients, i.e. with nifedipine and without nifedipine, was enabled by the increase in the number of initiating complexes in the couplon. So while increasing the number of ICs, or the number of calcium channels, decreased calcium release latency, the cost of this is that most of the calcium current is now not involved in triggering at all. So this plot shows this. The black trace shows total calcium current, and the blue shows the proportion that is involved in triggering. So as pointed out by Fabiato, this is the triggering portion of calcium current, and this, the rest of this can serve to load the SR. We can now also use these simulations to examine or to understand why calcium release variation may be smaller during the AP compared to, say, for example, RP. So this is sort of illustrated here. This figure F shows the evolution of P spark over time for AP in red and RP in black. And it shows that there's an excess of calcium spark production early on as well as later on. But during the AP, the early calcium sparks are prevented because the action potential peak is so positive, and this reduces calcium current and reduces calcium spark probability. Later on, the action potential is repolarizing, and this reduces LCC open probability so that fewer open and fewer sparks are activated. So to summarize, we found from our latency analysis that likely an initiating complex of 3 to 8 RYRs with 1 LCC can explain the calcium release latencies that we observed. And in the case of calcium transients, the latencies can be explained by increasing the number of ICs to 4 per couplon. And during the AP, P couple is 0.5, while during the calcium transient during the AP, P spark was near 1. So to achieve this robustness and the speed of EC coupling, the number of ICs needed to be increased, and this means that only 20% of the calcium current serves as a trigger for CICR. And to go through some implications.
Okay, thank you, Cherrie. So, so this is all, you know, quite hardcore biophysics. Perhaps does it mean anything that might be important? Well, I think in the context of the accumulating evidence by various groups, including a lot of work by Bill Lauch, that we've seen this cluster fragmentation under various pathological conditions. So what our modeling says that cluster fragmentation will alter the latency for EC coupling when the number of RYRs per initiating complex, remember that's the calcium channel plus the RYRs that it feeds, would be less than 3, and that we get less than 4 initiating complexes per couplon. So in the local area where we get the calcium spark produced, we have less than 4 initiating complexes. We're going to start to get an increase in latency and dispersion of release. And indeed, if you look on the right, there's an example of some changes, and you can see quite clearly the latency for calcium sparks are being altered in this system. And in the lower panel, some work, recent work we published with Ewan Fowler, who's now at Cardiff, we looked at a heart failure model. Now we know in that model some T-tubules are lost, so there isn't as many initiating complexes. But importantly, where we do get the release, the latency has increased, perhaps reflecting the sort of breakup of junctions. So any change in geometry may reduce coupling fidelity. And changes in L-type calcium channel gating alter latency when the closed time, the mean closed time changes and the mean open time decreases for whatever reason, like whatever drug or neurohumoral effects there might be. And the action potential itself will alter latency because if peak voltage changes, we change the relationship between the voltage that is activating the L-type calcium channel and the relationship between the consequent single channel current's ability to trigger a spark. And in connection with that as well, phase one repolarization, which we've spent quite a bit of time looking at, is also very important in determining release efficiency. A bottom line I suggest to you would be that changes in latency, which may be reflected in very non-uniform release, even if T-tubulars are still present, may actually produce intrasarcomeric stress because contraction time is going to be slightly different among sarcomeres. And that intrasarcomeric stress may somehow feed on through the cytoskeleton into some other changes within the cell. We just don't know what that means here. So in summary, thank you for attention. And I'd also like to thank the co-workers that we've had the pleasure working with in this study. Derek Lava for RYR gating data, some of which is unpublished. Laura Cannel and Andrew James here at Bristol, who've supplied us some cells to work on. And of course, funding bodies, importantly, the British Heart Foundation, but also some money from the MRC and others. And thank you very much, everyone.
Mark, thank you. And Cherrie, thank you very much for an amazing presentation. Hopefully people can hear. And we can now invite the participants to ask questions. If you would like to switch your microphone on and even your camera, it would be lovely to see you. Otherwise, you can pose questions in the chat, although at the moment, I can't see the chat. So Jenny and Mark, I don't know whether you can see any questions in the chat yet.
Okay, so Kaziak says, in this model, was the SR concentration considered? The dynamic change in SR concentration has a contribution to RYR open probability. Now, here we were considering the base case of a well-loaded SR. We haven't looked at what happens at other rates. So we've always paced the cell up towards steady state. And we assume the calcium concentration in SR was 1 millimole. That's right. Yeah. Nice talk. Luis Ganano. Nice talk. Do you have any comment about interspecies differences in spark latency? I mean, the relationship between heart rate and T-tubal density are a clustered arrangement based on your data AP shape. No.
That's a lot of work.
That's a lot of work. I think it would be interesting. I mean, we would pick the rat because we have here a very strong data set on which to actually firmly base our modeling. And we would obviously have to have an equivalent data set for an animal with a very different behavior, like, for example, the rabbit, where we know rabbit calcium current density is quite different. The action potential is quite different. So we would have to sort of kind of repeat this data. But in principle, it's eminently doable for other species if you get single cells. The techniques here would be applicable to almost any other cell.
The biggest gap is single channel L-type calcium channel data.
Yeah, that's really a big problem.
Great. Apparently, I'm told that we can't switch our cameras and microphones on. So sadly, we will not be seeing you in person, which is a pity, because there's a lot of familiar names out there amongst the participants. But if you continue to type into the Q&A, very happy to field any questions that you might have. Well, maybe I could ask you and Cherrie a question about pathology. I know you guys have done a lot of work over the years on the geometric changes that might occur in the dyad as a consequence of, for example, heart failure or whatever. Do you have any knowledge or predictions from your model as to how you could implement changes in geometry into the dyad that might occur in heart failure and how that's going to affect DC coupling? Is that the next step in the pathway?
I think yes. I mean, right now, you could tell us what geometry you want, and we can model it. I think the work being done by Bill Louch and others is going to be really interesting. I would like to see a lot more data on what the junctional SR looks like. As you may know, the previous paper we did JMCC, where we actually looked at high pressure freezing to actually get we saw a rather different picture of the junctional SR than I showed in the slides earlier. We actually have those samples stored from a heart failure model. And but we haven't processed them or analyzed them yet to see what the junction might change to in a realistic heart failure model. But I think Bill Louch's work has quite clearly shown this point, that there is fragmentation of the clusters. Although whether that fragmentation means that the single junctional SR stretches out and the clusters just get more dispersed, or whether the junctional SR breaks into smaller pieces, that I don't think is clear yet. So I think that's a really important question to feed into the situation. So I think we need more data. At this moment, all I can say is that our current understanding of EC coupling appears to be sufficient to explain EC coupling delays and voltage dependencies under normal conditions. The disease state waits further.
I think your model is super impressive in the way that it matches the data. So that's a great start. And as you say, you need more data to parameterize it for weird things like heart failure. Super impressive. I lost my sound there momentarily, but Eric Rios has got a question. I don't know whether you answered it already, Mark, about changes in mutations in RYR2 that affect latency. And do we know about any of the, can you model that in any way?
Yeah, well, certainly in terms of close time of the RYR, yes, that's quite achievable. I don't know, off the top of your head, how much of the total latency was the RYR close time compared to the? It was about 30% of the total latency. So, yeah, it's a bit under half the total latency is really due to RYR gating. So it's close time at those calciums generated in the diode. So I guess, I mean, I could sort of immediately tell you that, of course, if the latency, if the close time increases, you're going to add to that 30% latency to give you more latency. And you could kind of, just off the top of your head, sort of extrapolate. You see a third of it is due to RYR gating. Then you can sort of get a first order guess as to how long it might be if RYR gating does change.
Oh, well, I guess I'll just add that if in your experiments you don't see an increase in latency, it could be that it's actually just dropping off, like the calcium spikes aren't being activated at all. And so there's like an interaction of the two measurements. That's why we sort of had to use a model to help understand it. It's a bit confusing to think about it in your head. Can we answer Dan's question, or did we answer it already? He said, I was wondering, in the DP protocols, what were your holding potentials? -80. Can you rule out the effect of NCX while you're blocking sodium channel? We minimize the effect of NCX by having 0 sodium in the pipette. And the sodium channel was inactivated in these experiments via a ramp.
Excellent, thanks, Cherrie. Sorry, Dan, I missed your question in the Q&A. Feel free, anybody, to type more questions in the Q&A if they want. In the meantime, Mark and Cherrie, could I ask you again a species dependency question? And this is just showing my ignorance, really. You talk about, go back to Alex's Fabiato's estimations of 20% trigger versus 80% calcium loading. Is that likely to be the same across different species, or do you think the rat might be different there? Or what's the species dependence of the relative trigger contribution versus loading contribution? Any ideas?
Well, Alex, in a way, it's kind of surprising that Alex got this answer because, of course, his voltage if you remember the figure from his paper, I mean, he was thinking that the calcium current looked like the calcium current you see in response to a step. So it's like a current that activates really quickly and then exponentially decays. Of course, that's not what the calcium current looks like during the action potential. It's actually quite different to that. So the fact that he got, suggested that this might be the triggering component is sort of almost fortuitous. But the idea is right. Now, if you have a species that has a somewhat larger calcium current density and that relies more heavily on ICA versus SR, and Don Burrs has done some work on the species differences in terms of the amount of calcium supplied by the SR compared to the calcium current. So in rabbit, for example, the SR supplies slightly less calcium, we think. I mean, obviously in that species, the overall calcium balance shifts in favor of ICA. But in terms of the coupon itself, the triggering efficiency is sort of the same. So that then raises the issue of whether or not you've got more calcium channels that may not actually be in the coupon at all, perhaps in the surface membrane or somewhere else. Because if you've got a surplus much above what you need, and yet you have similar latencies, you've got to put those calcium channels somewhere. So once again, this comes down to better data, particularly immunolabeling and other things, to try and identify where all these calcium channels are. So I think Moore's work that suggested that a lot, if not most, of the calcium channels are in couplons or beside couplons, may be weighted towards being able to pick things up in immuno, where you need more calcium channels, in other words, more initiating complexes, in order to pick up the fluorescence.
But
So you may not just see singleton calcium channels lying around.
But Mark, your estimation of 8 to 1 is not a million miles away from, if I recall rightly, Don Burs's biochemical estimation of 7 to 1 or whatever it was, which would suggest there aren't any spare calcium channels hanging around.
Well, no, I mean, his 8 to 1, yeah, but that would assume that there aren't any spare RYRs either, and we know there are.
Okay, fair enough.
There are spare ones because it's called Corbular SR.
Fair enough. Yeah, I forgot about that. Excellent. No, very, very interesting. Not something I've thought about for a while, but excellent. Really, really interesting. We are approaching the hour mark here. If anybody's got any burning questions they'd like to post in the Q&A, very happy to pass them on to Mark and Cherrie. Just within the last 30 seconds, talking about the way in which the shape of the action potential and the square pulse has rather led Alex Fabiato to the right answer, perhaps by serendipity. Coming back to rabbits versus rats, the shape of the calcium current is, I don't know, but I assume it's significantly different because of the nature of the membrane potential and the action potential. Does that again give us another species difference, that the shape of the calcium influx is going to be profoundly different in those two species?
Yeah, I think it does. I think the rat is probably designed, I mean, we've been doing quite a lot of work recently on ITO, which is the key to getting this phase 1 repolarization. It's much faster and deeper in the rat, and also in the dog than in human or guinea. I mean, guinea pig doesn't have hardly any phase 1 at all. So this is a synchronization mechanism that decreases dispersion in timing of the calcium release process. So I guess rats and mice and things have really fast action potential, have really fast heart rates. They haven't got a lot of time to wait around. Maybe a few milliseconds becomes more critical at high heart rates. Whereas, of course, in larger species with correspondingly lower heart rates, you can wait a little bit longer for your calcium release to occur, provided you don't have too much dispersion in timing. And therein lies I think the key to the spike of the human or ferret or rabbit. There's a little bit of a spike in there. This really helps tighten up the timing. So it doesn't matter if you have to wait a little bit longer, as long as you don't get too much dispersion in timing, I suspect. But I think we'll have to understand what the consequences are. I mean, for subcellular remodeling and the sort of these models where the cytoskeleton feeds back onto gene expression, depending on what's happening to the cytoskeleton, I think is, a really interesting question, and might be an important thing to play on. I mean, is it that changes in the T-tubule, for example, that are known to take place in various disease states, does that lead onto the cytoskeleton, or does the cytoskeleton lead onto the T-tubule? And if so, what is it that's changing the cytoskeleton signaling that then promotes the changes in the T-tubules? So I think there's a lot of really interesting work to be done by lots of people to start looking at, signal transduction and how it progresses in the disease state. And I look forward to other people doing that.
I think that's an excellent way to finish, actually, Mark. In that I came into this assuming that you were going to tell us the answers to life, the universe, and everything. And what you've done is told us an enormous amount, but also raised lots more questions. And yeah, so you're not going to be out of a job for a while. I can see that. So,
all I can say is I know nothing.
Yeah, join the club. So fantastically impressive, Cherrie. Thank you so much for. The two of you, lovely piece of work and really thought-provoking presentation. Very interesting. Took me back to my youth to thinking about EC coupling as well. So thank you, guys. Just for the audience out there, thank you for joining us. If you've got questions for Cherrie and for Mark, I'm sure they'd be very happy to, when you've cogitated on your questions, for you to email them, and they will, I'm sure, be delighted to engage. So in the meantime, thank you everybody for joining this webinar. Thank you for JMCC and Jenny for hosting it, and particularly Mark and Cherrie for, well, a wonderful paper, but also a lovely presentation. And keep up the good work, guys, because it's very impressive. Thank you, guys.
Well, thank you, Mike. Thank you.
All right. Thank you, everybody. Goodbye. And I'm sure I won't see many of you before Christmas, so have a lovely holiday period, and hopefully, Mark, I'll see you sometime before the next 30 years. That would be great.
Well, I don't know. We'll see.
You're not so far away. All right. Take care, everybody. Bye-bye.
Bye.